Today we want to talk about protein structure. Proteins are made up of amino acids. 3 amino acids are shown here: aspartic acid, lysine, and glutamine. There's 20 naturally occurring ones. The key thing to note is an amino acid has an amino group, and this is the amine functional group in organic chemistry. It has a carboxyl group. This is the carboxyl group functional group, and both connect to a central carbon called the alpha carbon. Connected to the alpha carbon is in blue the side chains. All amino acids have an amino group, a carboxyl group, and connected to their alpha carbon are different blue side chains. Some polar charged, some polar uncharged, some nonpolar. Here I show the amino acids without their R groups, just designated by R1, R2, and R3 for simplification. Notice that the amino group can be charged or uncharged. The carboxyl group can be charged or uncharged. In this representation of 3 amino acids, I substituted in the subsequent representation an uncharged amino group here and an uncharged amino group here. Please follow the mouse pointers so you can see where I'm looking at. Well, this amine nitrogen with a lone pair, slightly negative. This carboxylic group, this carbon in the carbonyl, this is another functional group that's sort of a functional group found in many organic molecules, the carbonyl group. This carbonyl carbon, the C double bond O, is slightly positive. You can imagine if this amino acid got close to this, that this pair of electrons, a wannabe bond, might try to form a bond here, because that's slightly positive. Likewise, this might want to form a bond here. The net result would be this N on the second amino acid connected to this C in the first. And you can't have too many bonds to carbon, so the OH would come off, and also an H here is water, and you end up with this, which is a connection of the 3 amino acids, asp, lysine, glutamine, through the carboxyl group of the first amino acid and the amino group of the second to form a new functional group, the amide, which you should also know. So what we have here is now 3 amino acids that have come together. Notice the first amino acid has a free amino group. The last amino acid has a free carboxyl group. But that the carboxyl groups and the amines that were connecting the amino acids now are involved in an amide link, have no charge, and this is what they look like. So this is an example of what's called a tripeptide, 3 amino acids in which the monomer, an amino acid, gets covalently attached to another monomer and then another monomer to form what's called a tripeptide. Have a lot of these monomers connected together and you have a large protein. Notice that the protein has directionality. The first amino acid has an unreacted free amine. The last amino acid has an unreacted free carboxyl group. I've shown a larger peptide. This got 7 amino acids, but again, it shows you the same thing. A free amine N-terminus, a free carboxyl group at the end called the C-terminus. The amide bonds connecting each amino acids in the blue side chain shown. Again, they differ in their polarity. Now, this gets complicated for a big protein to show a protein fully with all its atoms, so we have sort of abbreviated versions. This is one in which I've taken all the side chains off and just replaced them with R groups, where the R groups correspond to the side chains you see above. We can even simplify it further by stripping off the R groups, and this would just show the backbone of the protein, or this small peptide. Something really important to note: almost every atom in this backbone is polar. Everyone except for, everyone, just about. So the overall background, you should look and find out which atoms are slightly negative and which ones are slightly positive, and you'll verify that. So we have a completely polar backbone, and then we have dangling side chains of various polarities, and we're going to figure out how that protein ultimately folds in 3-dimensional space to produce the final structure. The main chain, we can even simplify this further by just stripping off all the atoms and just having a line with an N-terminus and a C-terminus. Proteins have various hierarchies of structures. The primary structure of a protein is simply the linear amino acid sequence as we saw in the last slide. The secondary structure, something you've seen in biology, is when the protein backbone, protein folds on itself in repetitive fashion to produce repetitive structures, should I say, in which the repetitive structures are held together by hydrogen bonds within the protein backbone. For example, here's a protein backbone stripped of all the side chains, and notice I have hydrogen bond donors and hydrogen bond acceptors. If this amide hydrogen on this amino acid were to come close in space to this carbonyl oxygen, slightly negative lone pair of another amino acid further along the sequence, but close in space, you could form a hydrogen bond. So secondary structures are those structures, repetitive structures within a protein in which are held together by hydrogen bonds within the backbone of the protein. And we're going to see these include alpha helices, which are shown here in purple, and beta structure, which is shown in yellow. Here's an example of an alpha helix. We'll look at first at the amino acid sequence of this. If we're running in this direction, this is like the amino group. This is the alpha carbon and the C double bond O carbonyl oxygen, and this is the amino group of the next amino acid. The alpha carbon, carbonyl carbon of the second one. Notice this zigzag structure. I mean, this is when the protein backbone, that's what this is, it's extended completely. And we've seen that when we talked about alkanes. The zigzag nature in terms of being in the favored conformation. However, with a proteins fold on themselves, and when they fold on themselves, one of the things you can form is an alpha helix, and that's shown here in this little diagram with the yellows indicating hydrogen bonds. I'm now going to show you a rotating model of an alpha. This is an alpha helix taken from a protein, actually, from an Arctic fish. You can see looking down on it, this is what an alpha helix looks like in space filling mode. Let's simplify it. That's a wireframe view, and it's still not very simple because, frankly, it has a lot of all the atoms in terms of their bonds anyway present. Let's just show the backbone, and you can see that clearly the helical nature of the backbone. It's color-coded for the different amino acids. Let's throw in some side chains and take a look at them, and I think you can see that the side chains are all pointing, if you look down the axis, they're pointing away from the helix, okay? Now I'm going to show a ball and stick model with the hydrogen bonds. And these little dotted blue dashes are hydrogen bonds within the backbone of the protein side of this particular protein alpha helix. Finally, here's a cartoon model. And what's important here is, again, you see the propagation. The pink is just the backbone, the polar backbone propagating through space, coiled on itself. The blue represent hydrogen bonds. And please note again that all the side chains are pointing away from the helix. Another kind of secondary structure is beta structure. You saw that before in yellow. Take a look, for instance. I have a protein backbone. I've shown the backbone here running this way. And let's say it kept going this way and then came back on itself and continued in this direction. If they lined up in register so that a carbonyl oxygen could be really close on this part of the strand, could be close in space to the amide hydrogen further away, and they line up and register, we have what we call beta structure. This is just like alpha structure in a way, only it's a little bit more spread out. I'm going to show you a three-dimensional model of what that looks like. Here is a three-dimensional model of a beta sheet, actually, from a protein. Here's a wireframe model. And I think you can see I've cut the rest of the protein off, so you only see the chains, the polypeptide backbone running in these directions. Let's put ball and stick in a cartoon out, and now you can see the actual nature of the strands. They're all running in the same direction. So this is a backbone. A backbone, of course, this would propagate further in space. Now let's look at the hydrogen bonds, and there they are. In this particular case, I haven't added the hydrogens to the model. So here's a carbonyl oxygen, slightly negative with a lone pair. It looks like it's interacting with an amide nitrogen, but it turns out there's a hydrogen there that doesn't show up. And that's simply a hydrogen bond connecting the two. And here's the cartoon, again, showing the amide hydrogen bonds between the various strands. Proteins can also form tertiary structure. Tertiary, think of it as 3D. Tertiary, 3D. And this is an example of a protein. And you can see that there's alpha helix in this protein. This is just showing the protein, a single chain folded on itself. And there's beta structure here. And there's also this random backbone orientation. So not all of the protein is in the form of an alpha helix or beta strands. The final way to categorize protein structure is called the quaternary structure. And that's simply when multiple protein chains come together through intermolecular forces to form an aggregate, not a wild aggregate, but an aggregate of a specific structure. Here's an example of hemoglobin, in which I have 4 different chains color-coded in different colors. The proteins coming together, the individual chains can be the same protein or they can be different proteins. Finally, let's examine the question how proteins fold. It's complicated. We have a long backbone that's almost completely polar, and dangling off it are side chains. How is this thing going to fold in 3-dimensional space? Because the intermolecular forces that we talked about between 2 small molecules can also apply within a large molecule, in which one part of the molecule attracts another part in the process in which the molecule collapses into a final structure. Alpha helices are a form of this, in which you form secondary structure held together by hydrogen bonds between the backbone hydrogens on amide groups and the carbonyl oxygens. We've seen something like this before with the structure of my cell, which is an aggregate of lots of different fatty acids or carboxylic acids, in which we have a polar head group and a nonpolar tail. Let's remind ourselves of what that looks like. This is the structure of a micelle in 3 dimensions. We've seen it before. I just want to reiterate, if we I'll show one example of one of these groups. This has a polar head and a nonpolar tail sticking to the inside. If we were to look, where are all the polar head groups? They're on the outside. If we're asked, where are all the nonpolar tails? They are, as you know, on the inside. So this is a 3-dimensional spherical structure in which you got perfect separation of nonpolar and polar. Too bad that can't happen for a protein. When I say too bad, it's actually kind of fun because proteins are more complicated. Here's a question. If I have a protein that has a completely polar backbone and dangling side chains, which are connected side chain, where would a nonpolar side chain have a tendency to end up as the protein folds in 3-dimensional space? Let's take a look at a particular protein that I study in the lab. This is the protein tyrosine phosphatase that I study. It looks like a globular protein, not unlike a micelle, really spherical, 3-dimensional. What's the wireframe look like? And you can see it's pretty complicated. Let's look at the backbone. This is one protein collapsed on itself in 3-dimensional space, and you can see that the protein itself, this is just the backbone, winds in 3-dimensional space. Let's look at a cartoon model. I think you can identify the alpha helices and beta sheets, beta strands, which are running parallel here. Cartoon, we add a cartoon, you can see where all the side chains are. But the question is, where would a side chain that's nonpolar, such as phenylalanine, which is completely not polar, end up? Let's look. Well, I picked one. It seems to be buried completely in the center of the protein. Let's zoom in on it and see what might be around it. It's still pretty hard to see. So let's look at the individual amino acids that might be around it. There they are. Are they polar or nonpolar? Well, let's take a little bit closer look. And I have to slightly reduce the size of this molecule here after it stops spinning. And you can see that in general, this gray is all nonpolar, just carbon and hydrogen. It's surrounded mostly by gray. Not completely, but here's the sulfur here, and we know that's nonpolar. There's an oxygen here. You can't get completely away from having some polar atoms nearby, but predominantly surrounded by nonpolar amino acids. So, how does a protein fold? Really, we studied intermolecular forces. They occur between small molecules, big molecules. We talked about London forces, induced dipole, dipole, hydrogen bonds, and ion-ion. What about a protein? How does a protein fold? It's really application of intermolecular forces within a molecule to form this structure. On average, if you look at the bulleted points, half the amino acids in a protein are typically involved in alpha helices or beta sheet. The rest are sort of propagating in a random fashion. You would think that the location of side chains would vary with their polarity, just like it does for a micelle, in which not the side chain but the tail is completely buried. Turns out nonpolar side chains like valine, are predominantly buried in the interior of a protein. 83%. That means you can't bury them all. We talked about an example with hemoglobin, but almost all of them are. There's no way to do it with a protein because it's much more complicated. How about the charged polar side chains? charged and polar. You'd think they'd be on the outside. But it's a startling figure. About 54% of the polar side chains that are charged are buried. Typically, if they're buried with a partner that's of the opposite charge. Unpolar side chains, such as these amino acids, are mostly buried. Again, this is a little bit startling even to me as a biochemist. That's what the data shows after looking at the structure of lots of proteins. But it has to be that way because of the complexity of the protein. It's not so simple. That's why we started with lipids and understanding this. And ultimately, these globular proteins are compact. There's no water inside them. They're about as compact as a sodium chloride crystal.